In this video, we're going to talk about the quality assessment of newly assembled transcriptome of Rhinoptera javanica, otherwise known as the Flapnose rayfish. Key points. Here we built a new transcriptome reference for Flapnose ray, which is required for RNA sequencing analyses. The new assembly has a decent quality score based on following quality matrix: E90 and 50, BUSCO, and alignment score. These are the general assembly statistics, and what we want to particularly focus on is the E90 and 50. So E90 and 50 is a contig and 50, and it's based on the transcript contigs. So this is a transcriptome assembly, not genome assembly. So the contigs are not based on the chromosome, but it's based on the transcript, each transcript. And E90 means we only want to look at the top 90% of the expressed transcripts, which mean that we want to actually get rid of the bottom 10% of the expressed transcripts, which are lowly expressed and could be a bias or could be we're not really confident of them. So you can see the contig N50 is only around 500, but the unbiased E90 and 50, the relatively unbiased score is around 3000. And on the right side, you can see how this is represented. On the x-axis is a percentage expression, so it's from 0 to 100%. And on the y-axis is the N50 measure from each percentage expressed transcripts. And an empirical best case scenario, so this is empirically seen, and at around E90 you should see a peak, which means your assembly is relatively well done and you have enough sequencing depth to cover. And current project, what we could see is a kind of similar curve, and E9050, as I told you, is around 3000 kilobase. But the problem here is that we've got over 100% expression in some of the transcripts, and we don't know why this is, so we have to check this. This slide is dedicated to BUSCO score, so we have to look at the complete BUSCO score. And for complete, we've got around 99%, which is a very good score. It means that we have high identity orthogonal full-length homologs present in our assembly. And there's a concern about high duplicate over here in the dark blue. So complete but duplicated. And this is usual for the transcript because of the isoforms. So since this is a transcriptome assembly, so we've got a good BUSCO score from this assembly. This is the alignment score. So what we've done over here is to actually align the RNA reads back to our newly assembled transcriptome, and we had the two samples with RNA sequencing data. And we can see for each of them we've got 100% mapping. So 100% alignment achieved when mapping RNA reads back to assembled transcriptome, which means that we are safe to proceed with further downstream RNA sequencing analyses. Just one more to talk about, and this is a different topic. So this is a strategy for analyzing RNA sequencing sample when the DGE or differential gene expression study is not possible. And here is the problem. DGE is not possible when n equals 1. So as you could have seen from the previous slides, that we only have one RNA sequencing data from each case, left tumor and normal. And DGE packages such as DSeq2, will not run when n equals 1 because there is no variability that is required actually for the statistical study. And n equals, as I told you, n equals 1 means that variance is undefined. This is a solution. We're going to functionally annotate highly differentially expressed genes, and here the differentially expressed genes doesn't mean that we do any statistical studies, since I've told you that it is impossible. So instead what we're going to do is to just look at the simple subtraction. And by doing simple subtraction, what we're going to do is to actually sort them and only collect the highly differential genes over here. And we're going to do enrichment tests to see if they have any meaningful biology. And that was it. Thank you.